It's my pleasure today to introduce Dr. Kale Edmiston, who is a new member of our faculty who came last year. Dr. Edmiston received his bachelor's in cognitive science from Hampshire College in Amherst, and then got a PhD in neuroscience from Vanderbilt University in Nashville, Tennessee. He then was a postdoctoral fellow at China Medical University in Shenyang, China, and then was a postdoctoral scholar with Mary Phillips at the University of Pittsburgh Medical Center in Pittsburgh. He then went on the faculty at Pittsburgh and was an assistant professor in the Department of Psychiatry. And he's now an associate professor here at UMass Chan Medical School. He's won a number of awards and honors, too many to mention, but I'll just highlight a few. He's received a travel award in 2021 from the American College of Neuropsychopharmacology, which I can tell you from personal experience, it's highly competitive. He also received a Society of Biological Psychiatry Early Career Investigator Award and won the Research Day Department of Psychiatry Outstanding Poster Award. This is at the University of Pittsburgh, which I can also tell you must be highly competitive from that shop. He has a number of grants. He has a Kale 1 award from the NIMH and has received funding from the Brain and Behavior Research Foundation Early Career Award. He's the author or co-author of over 45 peer-reviewed publications and 5 book chapters. The title of Dr. Edmiston's talk is Novel Neurocircuits in Mood and Anxiety Disorders, Visual Limbic Networks. Let's give a warm welcome to Dr. Edmiston.
Thank you so much, Tony, for that kind introduction. I'm really excited to get to share a little bit of my work today with everyone. So my work in my lab really focuses on trying to understand the visual sort of bottom-up contributions to mood and anxiety disorders. So I have no conflict of interest to disclose. And the learning objectives for today, I'm hoping that by the end of my talk, you'll be able to describe the contributions of visual cortical networks to emotional perception, discuss visual perceptual differences associated with trait-based risk factors for mood and anxiety disorders, and 3, describe the potential use of visually targeted transcranial direct current stimulation, which is a neuromodulatory intervention, to reduce threat sensitivity in highly anxious individuals. But before I jump into all of that, I want to give a brief background about some of the theory behind how we process stimuli in our environment. So the idea here is that there are 2 systems that help to assign priority to environmental stimuli. And this is a critical and adaptive function because this allows us to prioritize our attentional resources in response to a changing environment. So on the left here, we have our automatic salience system, and this is a pre-attentive system that's associated with this automatic salience-driven orienting function. On the right side here, we have our goal-directed cognitive control system, and this is our effortful attention. So this is, when we use our cognitive abilities to focus on a specific stimulus in pursuit of meeting a goal. So these 2 systems must work in tandem. So for example, we need to be able to switch flexibly between them to meet the demands of our environment. So we may find that our automatic bottom-up process is associated with the salience of rewards in our environment. So if you have a lunch today, you may find that the salience of the cookie in your lunch is distracting and you can't pay attention to my talk. So your goal-directed control is not online. At the same time, this bottom-up stimulus-driven system is important for threat detection or potential threat detection. So if someone were to come in late and the door were to slam loudly behind them, we'd probably all orient our attention to the noise. But we should be able to switch back and pay attention to the talk again. There's quite a bit of trait-based variation in our ability to switch back and forth between these systems. So, for example, a highly anxious person might have a hard time disengaging their bottom-up salience processing network in response to a loud noise. It's thought that these two systems are actually mediated by different neural networks. So this is a schematic sort of representing a classic theory of how emotional stimuli are processed. So in this schematic, a stimulus is presented to an individual. It could be an emotional face expression. It's then identified and appraised for its emotional significance. This in turn produces an affective state, and then that affective state and our appraisal of it is downregulated in a top-down fashion. So much of the mood and anxiety disorders literature has really focused on that regulation component, but I'm really interested in better understanding that identification and appraisal component, because I think it's been overlooked in this literature. And I have some ideas about what might be associated with this process. So here's a network of regions that include the fusiform gyrus, the amygdala, and the anterior cingulate cortex. And this network is very important for threat detection in particular. The fusiform gyrus is an extrastriate cortex that exists at the junction of occipital cortex and ventral temporal cortices. And it's associated with the ventral visual stream and object identification, in particular face identification. But there's actually a broad literature suggesting that the fusiform gyrus tends to preferentially respond to any visual stimulus that we have expertise in. So for example, there have been studies of children who collect Pokemon cards, and they've been able to show elevated fusiform gyrus response in those kids. The fusiform gyrus shares reciprocal connections with the amygdala, and it's thought that these reciprocal connections facilitate the assigning of emotional salience to visual stimuli in our environment. The anterior cingulate cortex is associated with error detection, but it's also responsible for some regulation of the amygdala as well. So, as I mentioned, much of the mood and anxiety disorder literature has really focused on these prefrontal top-down regulatory functions. But there actually is evidence to suggest that there are visual perceptual differences in people with mood and anxiety disorders. People with these disorders tend to overgeneralize negative emotional stimuli. They tend to perceive threat more quickly than people without mood and anxiety disorders. And they also tend to perceive threat at a lower stimulus intensity. They also often show poor emotional discrimination. So these individuals might be more likely to interpret a neutral stimulus as negative or a novel stimulus as threatening. But when we look at the literature and the pathophysiological models for mood and anxiety disorders, they really do not incorporate any of this literature at all. They tend to be focused pretty much exclusively on prefrontal and limbic regions. And there's not much conversation about the bottom-up salience-driven processing in these disorders. So these are two recent figures from recent review papers, and you'll notice that there's no mention of bottom-up processing on the left, and on the right, the visual cortex isn't even visible. So they didn't think it was very important, I suppose. But when we look at the actual literature, there's quite a bit of evidence to suggest that there are alterations in visual cortical regions in mood and anxiety disorders. So this figure is from a paper that looked at individuals with social anxiety disorder and asked them to do an fMRI task where they were looking at a series of emotional faces. They were compared to a cohort of age and gender matched healthy controls. And they found that the fusiform gyrus bold signal was elevated during emotional face perception in the social anxiety disorder group. They wanted to look at how this part of the brain was functioning as a circuit with the rest of cortex. And so they looked at the coincident functional coupling of the rest of the brain, and they identified differences in fusiform coupling with the amygdala and the anterior cingulate cortex. So those plots are shown down here. Just to orient you, the social anxiety disorder group is the light gray bar, and the healthy control is the dark gray bar. And we see elevated fusiform amygdala coupling in the social anxiety disorder group, and the opposite direction of finding for the ACC. We see reduced fusiform ACC coupling. So this is consistent with a model where we have elevated bottom-up salience processing in our anxious group and reduced top-down regulation of that perceptual process as well. What about mood disorders? So somewhat similar findings in mood disorders. This is a meta-analytic review of the major depressive disorder literature, specifically the fMRI literature of emotional face processing. And they identified alterations in bilateral amygdala, fusiform gyrus, and also cingulate cortex, albeit a bit more posterior than the anxiety disorder findings. But of course, we know practically, for the clinicians in the audience especially, that we rarely see a presentation of someone who just has a mood disorder or just has an anxiety disorder. These things frequently co-occur. And this creates a lot of challenges clinically. We know, for example, that people who have co-occurring mood and anxiety disorders tend to have a worse illness course and more severe symptoms. It also creates some challenges for those of us who do research. It's actually very difficult to disentangle the relative contributions of depression and anxiety to brain function. So one possible approach to this is a study like this one. So in this study, again, an emotional face perception study, they looked at the bold signal response to emotional faces in 4 groups: a healthy control group, a major depressive group, an anxiety group, and then a group that had co-occurring depression and anxiety. And you'll see that fusiform and amygdala are, prominent components in all groups. But this doesn't really help us disentangle the relative contributions of depression versus anxiety. And the reason that that is so challenging is because these symptoms are so highly correlated. So before I jump into talking about collinearity and the challenge that it presents for us, I want to talk briefly about what collinearity is not. So in this figure, you might have your outcome variable Y, which is some brain measure, some functional measure. And you have all of your independent variables that might be different symptom measures. And so the yellow region represents the shared variance between X1 and Y. And this is relatively easy to model mathematically and doesn't really pose a big challenge for us. But you can do a similar thing, right, with depression scores. You could have a model where you had brain as your outcome variable, depression symptoms as your independent variable, and then this green area would represent the shared variance between them. You can do the same thing for anxiety disorders as well, for anxiety symptom severity. But of course, because they're so collinear, this creates challenges, because if we were to put both anxiety and depressive symptom severity in a model, we would really be modeling all of these green areas and the orange area. And so we wouldn't really be able to say, oh, this brain difference is due to depression, this brain difference is due to anxiety. Also, because they're highly collinear, this really reduces our power to detect effects. What if we could pull out that common internalizing component? So we can think of that as this orange region, the shared variance between depression, anxiety, and the brain. And that could be represented as a sum. We would also want to look at the specific component, the contributions of depression and the contributions of anxiety. And those are indicated here in green. And this would be a subtractive term. And so that's what we did in this study that I'm about to present to you. So we had our sort of expected correlation between anxiety and depressive symptom severity. And we Z-transformed our clinical scales, and then we summed them and subtracted them. And what this did was create 2 measures that are completely anti-correlated with each other. And this solves our collinearity problem. So now we can, in our model, include a term that represents the shared variance. So that would, we could think of that as a general internalizing symptom severity term, and the unique variance as well. So in this study, we had 43 individuals, adults with depressive disorders, and they were administered the Hamilton Depression and Hamilton Anxiety Rating Scales, which are clinician-administered scales. A diagnosis of a depressive disorder was confirmed using the SCID, and exclusion criteria included any history of psychosis or mania, or any substance or alcohol use disorder. All the participants were unmedicated at the time of their participation in the study. We then asked these individuals to complete an emotional perception fMRI task. So for this task, we used a set of stimuli from the International Affective Picture System, or IAPS. I don't know if people are familiar with this data set, but it's a large stimulus set of hundreds of images that have been rated for their valence and also their arousal. So that means that we have images that are neutral in valence. We also have images that are aversive in valence, but they could be low or high arousal. And the same thing goes for our positive valence component. Because the aversive component is of particular interest to us in mood and anxiety disorders, in this study, we just used the aversive and neutral image sets, and we selected images that had a social component to them. So all the images had a human social actor in them. Yeah, those are two examples of ones we used. Okay. So the individual would look at the image for 2 seconds. It could be a neutral or aversive image. And then they would view that image again for 8 seconds. If it's a neutral image, they're just instructed to look at it, just to attend to it. If it's an aversive image, there's 2 different possible instructions they might receive. One is to just attend to it, and the other is to reappraise their emotional response to it. And so the thought process here is that is going to be engaging that top-down regulatory function. Then individuals were asked to rate the strength of their emotional response. So all in all, for this task, there's 3 different conditions. As I mentioned, I'm very interested in the fusiform gyrus, the amygdala, and the anterior cingulate cortex. So those were our a priori regions of interest. I did something a little bit unique for this study, though. Most studies just look at the entire fusiform gyrus as the region of interest. But this actually doesn't make a lot of sense, because we know from postmortem human tissue data that there's actually quite a bit of variation in the cytoarchitecture and cell density across the fusiform gyrus. It's a very large region of cortex. We also know that there's a anterior to posterior functional gradient, as well as an anterior to posterior connectivity gradient. So to lump all of these regions together really isn't very precise. So we used a 4 subregion solution that's based on this postmortem human tissue data. So all in all, we selected 7 regions of interest. We have our bilateral amygdala, shown here in teal, our 4 bilateral fusiform subregion regions of interest, and then a dorsal anterior cingulate and a rostral anterior cingulate region of interest. We extracted bold signal during the task from these regions and conducted 7 parallel mixed model analyses, corrected for multiple comparisons. So our regional bold signal is our outcome variable, and we have, as a within subjects measure, our task condition. So that's the 3 different conditions in the task. And then also our shared variance between depression and anxiety. We can think of that as internalizing symptoms. The unique variance of depression relative to anxiety. And then the interaction between task and our 2 different clinical symptoms terms. And this is what we found. To orient you here, on the y-axis, I have my extracted bold signal. And on the x-axis, I have that shared internalizing component. So as we move further to the right on the x-axis, we have increasing severity of internalizing symptoms. And our primary significant finding was an interaction effect between bold signal in the anterior cingulate cortices and the task condition. So this effect is really driven by the negative correlation between bold signal and internalizing symptom severity during the emotional regulation component of the task in particular. So basically, people with greater internalizing symptom severity are less able to engage that top-down regulation function when they're instructed to do so. So I think, this isn't actually particularly surprising. We've known that there's reduced activity in anterior cingulate cortex in these disorders for some time. But I was also really interested in trying to understand how these regions were interacting with each other. And so to do that, we used a data analytic technique called generalized psychophysiological interaction analysis, or gPPI. This is a method that we can use to analyze task-based data. It's used to determine the coordinated activity of two brain regions. So to represent this graphically, we could plot out the timing of our task condition of interest. We can also extract and plot the bold signal from our seed region. And then we can extract the time course of the bold signal from our target region. We can then correlate them. And this allows us to show, to see how correlated the activity in two different brain regions is at a specific point during the functional task. It's important to remember that this is just a correlational measure, and I'll talk a little bit more in a minute about why that's important. So same regions of interest, but I selected the FG3 subregion as my seed region, and then these remaining 6 regions as the targets. The reason I selected FG3 is because of prior work that myself and others had done demonstrating that this portion of the fusiform gyrus shows the greatest increase in bold signal in response to emotional stimuli. So 6 parallel mixed models, similar to the previous mixed models in terms of the terms. So as I mentioned, these are just correlational measures. So we can't tell from this analysis what if FG3 is causing changes in activity in any of our target regions. It's just as likely that activity in, say, FG1 is causing changes in FG3. So this was a somewhat unexpected finding. I was really expecting to see fusiform amygdala, and we didn't. All of our findings were within fusiform subregion coupling. So again, to orient you, I have my functional coupling strength here on the y-axis. And on the x-axis, I have the HAM-D minus HAM-A term. So this represents the unique components of those two scales. A score that is more to the right indicates a person who has a more prominent depressive symptom presentation to their illness. And a score more to the left would indicate someone who has a more prominent anxiety symptom feature to their illness. So what we found is a positive correlation here, such that people with more depressive symptoms relative to anxiety symptoms are showing greater functional coupling in the fusiform gyrus. So this is really weird, right? We're not really sure what to make of this at first. It's pretty unexpected. But it does suggest that we can use the fusiform gyrus to distinguish mood and anxiety, or depressive and anxiety disorder symptoms. I will highlight that only this FG3, FG1 survived correction for multiple comparison, but it was actually the most robust finding in this study. So ACC bold is associated with internalizing symptoms. This is driven by the emotional reappraisal component of the task. Within fusiform gyrus coupling is associated with the unique component of depressive symptoms. And I hope that I've convinced you that the fusiform gyrus is the unsung hero of the threat detection network. But what about the mechanism? What do we do with this information? So what I think is actually happening here, and this is, I will say, very speculative, is I think that the within Fusiform Gyrus findings are associated with predictive coding. So in order to accurately perceive our environment, we depend on a set of prior expectations that are based on our past experiences. These environmental regularities are transformed into predictions after the current sensory signal is processed. And so here in this figure, we have our population cells, which are the representational cells, and E population cells, which are error cells. And these prediction error cells back project to our representational population to fine-tune the neural representation of a specific stimulus. The idea being here that we're trying to minimize the mismatch between our expectation and reality. So this process is why, if you were to view the same thing, like in a scanner, if I were to show you an image of the same face over and over and over again, your visual cortex response would gradually decrease over time. You would habituate to it. But we know that in anxiety disorders in particular, that doesn't happen. These individuals do not habituate over time. This, coupled with the within Fusiform Gyrus finding that I've shared with you, leads me to think that there's some issue with the predictive coding that's happening in the visual cortex for these individuals. And this could explain why there's an overgeneralization of threat in mood and anxiety disorders, and why people tend to misinterpret novel and neutral stimuli as threatening. So we have a circuit. It's really focused on Fusiform Gyrus and the anterior cingulate. And what we've demonstrated is alterations in the Fusiform Gyrus within functional coupling and reduced anterior cingulate cortex projections. So can we actually modulate this circuit as a potential intervention? So that's what I'm working on now. I'm using a model called Transcranial Direct Current Stimulation, or TDCS. This is a neuromodulatory technique that involves application of a very small current to the scalp via two sponge electrodes, an anode and a cathode. Unlike other neuromodulatory mechanisms, TDCS actually uses a steady state current, so there's no alternating current. And the mechanism by which it acts is it's thought to change the polarity of neurons. When I explain this to the participants in this research study, I describe it as turning up the volume or turning down the volume on the brain. So the cortical regions that are under the cathode tend to hyperpolarize. And so therefore, during active TDCS, they tend to fire less. In contrast, the cortical regions under the anode depolarize, and so they tend to fire more. This is a relatively new method, but there has been some literature to provide guidance in terms of the duration of stimulus that's appropriate, as well as the intensity. So this is a study where they looked at motor cortex excitability with TDCS. The top figure shows different intensities of TDCS, and the filled in circles are the intensities that resulted in a motor cortex excitability that was different from baseline. So what we can conclude from this is that really 1 milliamp of current is ideal. This figure used for this experiment, they used 1 milliamp of current at different stimulus durations. And again, we see that you really need several minutes of stimulation to affect cortical excitability. Much of the TDCS literature is really focused on unipolar depression, and it's focused on targeting frontal regions. So these are two classic montages that are often used. The red sponge electrode is the anode, the blue is the cathode. So this is targeting the front part of the brain, and the current flows from the anode to the cathode. But because of my interest in the fusiform gyrus and the anterior cingulate cortex, I've opted to try a novel montage. So this montage has the anode right over CZ, which is right here at the top of the head, and the cathode at OZ, so right here at the back. This is a computational model showing the relative strength of the electrical field with this montage. And you can see that the strongest electrical field is really targeting visual cortex, as well as anterior cingulate cortex. So I conducted a small pilot study where we recruited adults from the community, just 10 individuals. And instead of focusing on a specific disorder, I actually opted to recruit people ranging continuously in trait anxiety. And that's because trait anxiety is a transdiagnostic risk factor for almost every psychiatric disorder, but particularly mood and anxiety disorders. People with high trait anxiety tend to view the world as more dangerous or threatening than those with low trait anxiety, and they're more likely to respond with an elevated state anxiety response than people with low trait anxiety. So we use the Spielberger Trait Anxiety Inventory to assess trait anxiety. This is a 30-item Likert self-report scale with scores that range from 20 to 80. And a score greater than or equal to 38 is considered moderate to high trait anxiety. It's important to have individuals complete a task while they're doing TDCS. And that's because we're modulating the activity of a specific circuit. So we want to make sure that we're having people engage in a task that would use that circuit so that we can moderate the activity of it. And so for this study, we asked people to complete this threat sensitivity behavioral task. So this is a block design task. There's 2 blocks of faces. And the face is it's a short video that morphs from a neutral face to either a happy or a fearful face. So that looks like this. It's a little over the top. So it's a 4-second video. And we ask people to press a button when they first perceive the emotional expression. So the idea here is that an individual with high trait anxiety would perceive the threat at a lower stimulus intensity. So they would press the button earlier. And so that is our measure of threat sensitivity for this task. So like I said, there's 2 blocks each, 2 blocks of happy, 2 blocks of fear. There's 30 trials in each block. So there's 60 of these trials for each emotion. So we asked people to come in for their first visit. They completed a very comprehensive clinical and trait personality assessment. And then for visits 2 and 3, they completed either an active or a sham TDCS session with a 24-minute face morph task. So this was a double-blinded study, and the order of the active versus sham was randomized. So these are individual subject data from our happy face trials. So I said that we recruited 10 people. You'll notice right off the bat that there's only data for 6 people. And that's because what I discovered was that this task was really long and boring, and people fell asleep during it. So lesson learned, 24 minutes of button pressing. It's too long. So there's some missing data. We had one person who opted to quit in the middle because she found the TDCS uncomfortable. So we only had one person drop out due to tolerability issues. In any event, these are our individual subject data. So we have our button press latency here on the y-axis. So a greater button press latency would indicate less threat sensitivity. In green is our sham, and in blue is our active. And I've sort of outlined here in blue subjects 2 and 4, who are our high trait anxiety participants. And the good news is that there was no effect of active versus sham on happy face perception latency. This is great because it means, this is our control condition. We're not affecting people's visual acuity. here's the data from our fear face trials. And again, you'll see 2, 4, and 6 are high trait anxiety individuals. And it's a small sample, it's very preliminary, but I did decide to go ahead and look and see if there were any effects of active versus sham on button press latency. And there was. So basically what we were able to find was that the active TDCS condition is associated with reduced threat sensitivity. We're really underpowered to see if this is specific to high trait anxiety. So that's my next project. So what I'm hoping to start recruitment for very soon here is a project where we're going to expand the sample and use a very sort of similar montage and model. Again, people will come in for their first clinical trait and trait assessment. And then they'll come in for 2 more visits. The difference, though, is that we're actually going to be able to conduct the TDCS sessions in the scanner while we collect fMRI data. This is very exciting because not only then do we have this behavioral outcome, but we can also look at how the active TDCS condition modulates the brain in real time. You'll also notice that I learned my lesson and I made the task quite a bit shorter. So hopefully no one falls asleep. So I'm hypothesizing that the active TDCS condition with the cathode over visual cortex is going to reduce bold signal in visual cortex. I also hypothesize that it's going to increase bold signal in the anterior cingulate cortex and reduce threat sensitivity by increasing the button press latency. But with a larger sample, I can also look at moderation effects. And so I'm going to look at moderating effects of trait anxiety. My hypothesis is that the effects on ACC and threat sensitivity are going to be greatest in the most anxious individuals. So I'm really excited, looking forward to launching this project, hopefully in the spring. So I started out today with a very simple circuit, 3 brain regions. But of course, we know the brain is much more complicated than that. And our threat detection network includes a lot of brain regions that I don't think get their due attention in the human subjects neuroimaging literature. So I'm really excited to try and look at some of these other regions, in particular the colliculus, which is a nucleus that's responsible for our head turning orienting response. So that's a bottom-up salience processing type region. I'm also really interested in looking at some of these thalamic subnuclei. And finally, also the basal forebrain. I also think it would be really cool to look at different sensory modalities. So I've really just focused on the visual system, but I would expect that this would be a domain general phenomenon that would also occur in other sensory modalities as well. Finally, I'm also interested in thinking a little bit about the developmental angle for some of these regions. So a lot of these bottom-up salience regions associated with visual cortex develop earlier than prefrontal cortex. Anxiety disorders tend to have an earlier onset than most other psychiatric disorders. And so it's possible that these bottom-up visual perceptual networks are altered early in development. And that sort of sets people up for a developmental trajectory where they have alterations in these downstream prefrontal regulatory regions. So I'm really excited to maybe look at some of these developmental effects and eventually see if there's some sort of neuroplasticity type remediation that we could do. So I have my work cut out for me, but I'm really excited to do it. So with that, I'll thank you all for your attention. And I want to highlight a few people in particular. I want to highlight Mary Phillips, who is my K01 mentor. Jay Fournier, who is a professor at Ohio State, and it's his K data that I presented in the first part of the talk. Tiffany Nan, who is the research coordinator here at UMass, who's going to be collecting all of this fMRI TDCS data. And then Christy Mack, who is the research assistant at Pittsburgh, who's been working with me since she was an undergrad. So She made all of those face morph videos, which is a very tedious task, and collected all of that pilot data. And of course, I want to thank my funding sources, and now I'll take any questions. Thank you.
Hi, thank you. This is really cool. And as a neuropsychiatrist, I'm kind of like into this type of thing. I was thinking about, you presented a piece of your data, the particular slide where you said this was somewhat surprising and all of the connectivity was just in, that one Gyrus. And then you said, maybe I misheard, but do you think that it's a unilateral finding? Because you said only these regions really panned out when you tested for the multi-test, kind of hypothesis. Do you think this is purely a left-sided or right-sided?
Oh, all the regions of interest were bilateral. So I thought about looking at laterality, but with only 43 people in the sample, I didn't want to overfit the model. But that would be a really important future direction because there are some laterality findings with fusiform gyrus for sure. So
thank you.
Yeah.
Well, I just wanted to make a comment / question. I mean, you know this, but I think everyone knows this, but this whole issue of anxiety and depression, the tremendous overlap between and many people in the field kind of think they may have this, they may be the same manifestations of the same disorder. Is your work hoping to shed some light on that? Because I mean, I know you're taking great pains to separate it out, but I'm just wondering, it seems like this work would shed some light on this.
Yeah, I mean, I think that's one of the possible aims is, if we can show that there are brain regions associated with difference, the unique components, then that would indicate that they're not the same sort of underlying pathology. I tend to think that there's quite a bit of overlap, in terms of thinking, internalizing symptoms generally, but I do think there's something different going on. I mean, this is pretty speculative, but particularly when we think of sensory processing, we often think of depression as being more internal, and perhaps anxiety disorders are more reactive to external stimuli. So that could be one potential mechanism by which, we could differentiate them.
Yeah, I mean, I'm thinking of the pharmacologic treatment of these disorders. These medicines we call antidepressants work in anxiety, and it's just very, very interesting to think about that they could have the same underlying pathophysiology, and just the phenotype is manifesting in different ways.
Yeah, and I think that mechanism of thinking about the predictive coding could be really important there, because a lot of that is done via lateral cortical connections that are, more serotonergic or, noradrenergic. So that would fit with the treatments that we have.
Right. Other questions? We don't have any questions online.
Oh, wow.
Thank you. Thank you. Thank you very much.
All right. Well, we get out of here early. Thanks, everyone.